This is DNA structure and gene expression tutorial presented by Dassi SchrDbb with reference ABC of Clinical Genetics by Helen Kingston. Hello, so from here we're going to start with the DNA structure and the RNA structure. So we start with the first introduction. You need to know that what the DNA is made up of and the RNA is made up of nucleotide. Another name of DNA is also called deoxynucleic acid, deoxyribonucleic acid. Another name of the RNA is also called the ribonucleic acid. Now, all of them are made up of nucleotide, and the nucleotide is made up of three main elements. So you first have one element here. So this first element here is called the sugar base. So this is the element of the nucleotide. The first element here is the sugar base. The second element here is going to be called phosphate linkage, and the third element here is the nitrogenous base. Now, the sugar base at the level of the C1 of the sugar base, that is the carbon number 1 of the sugar base, this attachment of the nitrogenous base. At the level of the attachment of the carbon number 5 is the attachment of the phosphate linkage. Now, the difference between DNA and RNA is that the first difference is that deoxyribonucleic acid is deoxynucleic acid is made up of the oxygen at the level of the carbon number 2 of the oxygen at the level of the carbon number 2 of deoxyribonucleic acid DNA. There is a removal of oxygen here, while ribonucleic acid is made up of ribose base. So the sugar base here, since the oxygen at the ribonucleic acid is not removed, the sugar base of ribonucleic acid is called ribose. Why, since the oxygen base of carbon number 2 of DNA has been removed, the sugar base of DNA is called deoxyribose. Now, DNA and RNA also consist of nitrogenous bases. As you can see here, we have names, acronyms. T is for tyrosine, A is for adenine, C is for cytosine, and G is for guanine. So these are the different nitrogenous bases that are found at the level of the DNA, and they bind to complementary to the different nitrogenous base. Thymine is going to, not tyrosine, sorry, thymine is going to bind with adenine with a double bond, and cytosine is going to bind with guanine with a double bond. Now, we need to know the difference still between DNA and RNA is that there is no thymine in DNA, but there's instead uracil. So this is thymine, which is replaced by uracil at the level of the RNA. Now, this is just what is called DNA replication. Now, DNA replication, you need to know that DNA replication usually occurs from the 3' to the 5' direction. When I say 3', I just mean the part which is closest, which is at the level of the carbon number 3. You see that here is carbon number 1, carbon number 2, carbon number 3 of the sugar base. Is it clear? So this is the sugar base, carbon number 3. So this is a 3' position. And now here is carbon number 3, carbon number 1, carbon number 2, carbon number 3, carbon number 4, and there's another carbon here to which the phosphate bind, which is called carbon number 5. So that's why it's called 5' position. So from this position here to this position is from 5' to 3', and from this position to position is 5' to. So from this position here is 3' to 5' direction, while from here to here is 5' to 3' direction. Now, you need to know that generally DNA replication occurs from the 3' to the 5' direction, Why? So as you can see here, so it's going to be occurring in a continuous pattern from a 3' to a 5' direction, because 5' are going to be produced here, and then the 5' contain the phosphate base, which is like an initiating factor for the DNA replication. So DNA replication start at the level of the phosphate base, because the phosphate base is like an initiating factor for the reaction to occur, and then for the replication to occur, and then it occurs now in this direction. So this one is from the base from which it is replicated, from which the DNA is replicated, is going to is 3' to 5'. Why the base which is replicated is 5' to 3'. Are you understanding? In order to produce a complementary pair. Is it clear? Now this other line, you need to know that this is going to be continuous when it's produced from 3' to 5' direction, and is going to be discontinuous when it's produced from 5' to 3' direction, because it's going to come forward while going backward at the same time. Now transcription now is a conversion from the DNA to the RNA. Is it clear? So when you want to convert from DNA to produce messenger RNA, you do what is called transcription. Now transcription here is what occurs in this case. You see, transcription also occurs in the same direction as DNA replication. It occurs in the 3' to the 5' direction based on the DNA base. Is it clear? Let's say that this is the DNA base here, from which it is going to be transcribed, and this is the mRNA. So you see, this direction is 3', so this direction is going to be 5'. So this is 3' to 5' direction. You see that this segment of mRNA is going to be complementary 5' to 3' direction. Is it clear? So as it comes here, is going to be producing the complementary linkage. Guanine is going to bind with cytosine, guanine cytosine, thymine adenine, cytosine guanine, guanine cytosine. But you need to know that while on RNA there is no thymine, so adenine, uracil instead replaces thymine, so adenine is going to have a complementary base pair with uracil and not thymine on RNA. So now we need to know that what after the production, so the chromosomal DNA can produce the messenger RNA, which is the mRNA. It can produce the ribosomal RNA. The ribosomal RNA is what is going to be constituent of the ribosome. Is it clear? And it can also produce the transfer RNA, and the transfer RNA is what is going to be used in the production of the protein product. And also have the snRNA, and the snRNA is what is going to enter back in the RNA transcript. Now genetic code, you need to know that what the genetic code of the human body is coded within 3 nitrogenous base pair. Is it clear? So all the proteins, each amino acid in the body, there are 20 amino acids in the body. There are 20 amino acids in the body, but there are 64 possibilities for you to have codons. In order to have all those 64, so there are 64 possibilities for you to have codons. Is it clear? But there are so there are 64 possibilities for you to have codons, but there are only 20 amino acids which are being coded. It means that codons which are base, which are additions of three consecutive codons are actually addition of 3 consecutive nitrogenous base pair. Is it clear? And these 3 consecutive nitrogenous base pair code for 1 amino acid, and there are certain codons which are stop codons, meaning that what immediately that those codons are going to be formed, there is going to be stoppage of replication. Is it clear? So this is an example of a codon, or stoppage of transcription. So an example of a codon, so you can see here U and U is going to code for phenylalanine. Is it clear? U and C is going to code for phenylalanine. U and A is going to code for leucine. So those are a certain example. So C, U, and U is going to code for leucine. C, A, and U is going to code for histidine. Now G, C, and U is going to code for alanine. Is it clear? So from this table you can have all the codon sequence, and this part here is from the 5' terminal to the 3' terminal. So now it's from 5' to 3' terminal, because don't forget that in this initial diagram you need to know that the replication of the DNA is from, and it's the transcription of the DNA and the translation of the DNA is from the 3' to the 5' direction, basically. But we need to know that what, since transcription resulted to the production of an mRNA which is in a 5' to 3' direction, when you are doing translation of this mRNA is going to be in a 5' to 3' direction, and this still correspond to a 3' to a 5' direction in the DNA. Is it clear? So now let's go now to the next one, which is translation. So for translation to occur, we have what is called the tRNA or the transfer RNA. Is it clear? And that transfer RNA has anticodons. Is it clear? So there are anticodons that are going to pair with the different codons on the transfer RNA. And then at the terminal portion of the transfer RNA, we have the different amino acids which are being coded. So let's take an example. CCA. CCA is a codon. The anticodon which is found on the transfer RNA is GGU. But this anticodon is not what is going to be used in the production of proline. No. What is used in the production of proline is CCA, the codon. So let's go back in the diagram up here to find CCA. So C, C, and then A. You realize that it is proline actually. Are you seeing? You're seeing that proline is going to be coded from CCA. Now the next one is GCU. GCU is coded by what? So let's go. We see G, we see C, and we see U. GCU is alanine. Effectively, on this diagram down here, GCA is coded. I said that you don't take this one here. This one is the anticodon. It's not what is going to be coded to produce the particular protein. The coding of the particular protein comes from this, which is GCU. And going back to find GCU, you have alanine, which is effectively this. Is it clear? So don't use these symbols to find for the amino acids. No. Use instead these symbols on which are the codons on the mRNA to find the amino acid, and the codons on mRNA for the amino acid should go in a 5' to a 3' direction. Now the next one is proinsulin. So those are these. So we need to know that this is actually how genetic coding goes. Is it clear? Actually, when you have gene translation, so for you to have gene, when an mRNA is produced, let's say this is a DNA. The DNA consists of different parts. You have an exome, the exome 1 portion, exome 2 portion, exome 3 portion, intron 1 portion. You have exome 2 portion, you have intron 2 portion, and exome 3 portion. Now the exome portion are the portions of the DNA which are going to be expressed. Is it clear? While the intron portions are the portion of DNA which are not expressed. Is it clear? So that's the difference. Exome portion are the portion which are going to be expressed, while the intron portion are the portion which are not expressed. Is it clear? Now at the terminals of the DNA, you are going to have the untranslated portion. You have the 5' untranslated portion, and you have the 3' untranslated portion of the gene. Now, at the level of the untranslated portion, at the end of the untranslated portion, we have stop codons, such that they stop to translate. So that they stop to transcript. Now these are the different caps. So on the mRNA, you still have the exon portion, the intron portion, the exon portion 2, the intron portion 2 and the exon portion 3 with a different 5' translated portion, the terminal part of the mRNA. Now what happens is that there is going to be an enzyme inside the enzyme inside the nucleus is going to split the messenger RNA. Is it clear? Such that it's going to split the messenger RNA such that it's going to excise the intron portion, because the intron portion are not going to be translated. They are made up of heterochromatins. They are not going to be translated. So there's excision of the intron, splicing exons, and polyadenation. So polyadenation means that what they are going to be terminal heads here. That is the polyadenation. Polyadenation means that the terminal heads, the head and the tail, are going to be the mature mRNA. So we have the exon portion in the center, and we have head, and the head is with the cap of the 5' M terminal, and then the tail is here like that with polyadenation. Is it clear? Now the next one. So after that, now we have this, which is polysome formation. So at the level of the ribosome is going to produce polysomes. So when it's passing at the level of the ribosomes, we have the amino acids that are being linked by the peptide bonds, and when the amino acids are linked by the peptide bond, there is formation now of a protein, which is going to result to a formation of an enzyme which can now interact on biomolecules to produce the cells, and then which interacts now all through in the human body to produce yourself. So that is why the human body is self, as you can see. Now then we have the genomic organization. Genomic organization, you just need to know that the DNA is made up of a double helix. After that, we need to know that the double helix is going to wind on certain, you're going to make a 2-turn round winding on the certain protein, and that protein is going to be called the histone protein. At first, it was thought that the genetic material of a human body was stored inside this protein called the histone protein, but later on, Watson and Sir Crick have come out with a theorem that tells that the DNA is the one that is storing the genetic material. Then later on, this is called the nucleosome, when the DNA is encycling the histone proteins. Then later on, now the nucleosome now forms multiple groups. They produce the chromatin fiber. Then the chromatin fiber in multiple groups produce loops of chromatin, and then loops of chromatin in multiple groups produce chromosomes in metaphase. Actually, if the chromosome is in prophase or also in interphase, they exist like chromatin fibers like this. When they are in interphase, when the chromosome is in interphase or it's in prophase. But when in prophase, the chromosome exists as loop of chromatin, but in interphase it exists like chromatin fiber. At the level of metaphase now, the loops of chromatin are well contracted now to produce this big chromosome in metaphase. Is it clear? So from here we have visualized all what was involved inside this tutorial on genetics. So we say thanks for your kind attention. Please don't forget to like and subscribe for our channel. Thanks very much.